Hello, everybody. First, I would like to thank for the invitation. My name is Aliyah Wang. I'm a principal scientist working at Pfizer in St. Louis. Today, I'm going to talk about advancing biotherapeutics characterization with ultra-high-resolution mass spectrometry. Here's the outline of my talk. First, I'm going to talk about the role of mass spectrometry in Pfizer's biotherapeutic heightened characterization strategy. Then I will give two case studies, data collected on Bruker maxis Q12 for in-depth understanding of biotherapeutics from both intact and subunit level. Then I will switch the gear to the peptide level analysis, talk about degradation hotspot, including Asparagine deamidation and aspartic acid isomerization. Our is mass spectrometry and biophysical characterization. We are analytical research and development within biotherapeutics pharmaceutical sciences. We primarily support development track starting from the early development all the way to phase 3. There are two major focuses in our group. The first is to apply state-of-the-art technologies for in-depth structure characterization, and second is to investigate new instruments, technologies, and methods. Pfizer has a very diverse biotherapeutic portfolio, ranging from less than 20 kilodalton to all the way more than 500 kilodalton. Here, I'm only showing a very limited examples of the commercial products. To support such a diverse portfolio, versatility of the instrument is critical. Pfizer has a very diverse biotherapeutic portfolio. Here, I'm only showing a few selected examples in the commercial space. It ranges all the way from less than 20 kilodalton species, such as peptides, oligonucleotides, all the way to larger than 500 kilodalton therapies, such as glycoconjugates or gene therapy program. To support such a large, diverse portfolio, versatility of instrument is critical. Here I'm showing you Pfizer's heightened characterization strategies by mass spectrometry. It's a tiered top-down strategy from intact analysis in which we get the entire molecule without further processing, to subunit analysis in which we will chop the molecule into 2 or 3 parts, all the way down to peptide level analysis in which we will characterize set-specific attributes. These complementary mass spec characterization techniques will ensure good product and process understanding. To support all those methods, we use maxis Q12. This is the maxis 2, and it is the workhorse for all the platform methods in our lab. What makes this instrument really attractive to us is it has ultra-high resolution, easily more than 80,000, and a really good mass accuracy. We got less than 2 ppm in a routine way, and also a very wide dynamic range. So we could support all these different assays on a single instrument. We also have a few other Bruker instruments in our characterization lab. Here I'm showing you not only mass spec but also a few NMR instruments. With mass spec, we have 2 maxis 2 installed in 2016. We have just a new one purchased and running right now in 2019. We also have older generations of the maxis Q12, Impact 2, and also a multi-12. And with NMR, we are very excited that we got the new this year, an 800 megahertz advanced NEO with cryoprobe, and also we have another 600 and 300 in the lab. So the way we see our future is we are trying to do characterization from both mass spec and NMR, and we are really looking forward to more sparks between these two methods. Before I talk about case studies, I want to emphasize that even we are a mass spec lab, separation is a key consideration for method development in our lab. Here I'm showing you a few different categories, and we are trying to build a toolbox of separation methods to expand our capabilities. For the subunit and the peptide, reverse phase chromatography is the most commonly used one. And we will use size exclusion for intact and also assembled molecules, including higher molecular weight species and also heterogeneous molecules. We also look into use like interaction chromatography to study either mono, oligo, polysaccharides, unit analysis, and protein analysis. In the recent past 2 years, we added a charge variant ion exchange chromatography coupled to mass spec, and we developed both intact and subunit analysis. There are also other chromatography methods we are developing, such as hydrophobic interaction. There are also affinity column methods we are looking into. Of course, there are non-chromatography-based separation. And actually, today I'm going to talk about ion mobility as well. So here, I just want to re-emphasize the point that separation and the mass spectrometry are very powerful for this characterization. So I'm coming to my case studies. So first, I'm going to talk about the data collected on the maxis Q12 for in-depth understanding of biotherapeutics. Additionally, we perform three-part subunit analysis, which means we will utilize enzyme to cut at the hinge region, and also followed by reduction to generate this three-part light chain FD and a single chain FC. So now you can see there are three parts. However, the challenge with the ice digestion is it can only cleave mAbs. For example, with the AAA mutant, it won't work. And there are a few alternative enzymes. In our experience, they are not as specific as ice. They tend to have more artifacts. Recently, we have been transitioning our subunit analysis from three-part to two-part, which means in this approach, we will only utilize reduction. The advantage of the approach is it's applicable to all mAbs, and we don't need to worry about the AAA mutant. And also, it has a simplified sample preparation, and of course, we can eliminate digestion artifacts. This transition is made possible because there is enhanced resolution on the maxis instrument. Now we are able to obtain monoisotopic resolution on both heavy chain and light chain. And I'm going to show you an example here. So on the left is a experimental data on the light chain on the top. This is deconvoluted data. On the bottom is the theoretical simulation of this light chain. So you can see the isotopic, not only is isotopically resolved, but also it matches very well between experimental data and the theoretical, and the mass accuracy is less than 1 ppm. Region with its almost 50 kilodalton, if you focus on following my curves here, we also monoisotopically resolved the heavy chain, good matching between theoretical and observed, but also for a modification which is at a much lower abundance. This is the C-terminal amidated proline on a heavy chain, which is 50 dalton difference. So you can see we also get a monoisotopic resolution, and it's less than 1 ppm mass accuracy. So here this data is showing that this high resolution data reads as confident identification at the subunit level. And because this is only reduction only, it is a fairly quick analysis. Now I'm going to talk about intact analysis on maxis 2 Q12, and I'm going to talk about the trisulfide rapid quantification. So trisulfide is a common PTM occurring in IgGs, and this carton is the IgG1. So for IgG1, this additional sulfur typically occurs between the heavy chain and the light chain. The root cause of trisulfide is shown to be related to cell culture conditions. This is our deconvoluted data at the intact level. Now you can see the most abundant species is our intact mAb, and this little bit shoulder trisulfide is plus 32 Dalton, and this is really good. Although this is not baseline resolved, the fact that we are able to detect this trisulfide at the intact level is really helping our analysis. Now we can easily quantify this trisulfide, use the intensity of B over A plus B, get about 20.4%. And this matches very well with the peptide level analysis, which typically takes much longer time. Because of this ability, now we are able to go through a lot of samples to look at the trisulfide analysis. And one example, how do we use the intact analysis to do the trisulfide screening for bioprocess? Bioprocess support including many types of screening, such as days in the culture, clones, different growth conditions, or media selection. This example is about the days of culture. So starting from the top, we have G0F with G0F, the major glycol form, and the plus 1 trisulfide plus 2 trisulfide. And at day 7, you can see we have about 48% of trisulfide. And in a similar way, at day 9, 41, day 11, 36, it starts decreasing as the days in the culture increases. And at day 14, there is no longer detectable trisulfide. So in this way, we can give a really fast readout to our partner from the bioprocess group to help them design the experiment. So now I'm going to switch to talk about data at the peptide level about degradation hotspot. To support our medicine design at Pfizer, we have this initiative a few years ago to look into the hotspot. So the goal of this team is to look into almost 100 Pfizer mAbs, and we will stress them under different conditions, try to build a database around this data set, and continue building a predictive model. So this way, in the future, we can ideally design a medicine without potential hotspot. Here is our workflow. We will stress the molecules at 4 weeks at elevated temperature, different pH. We utilize different pH because we want to make sure all the potential hotspots can be induced under all conditions. And we will typically use imaged capillary electrophoresis, which is shortened as ICE, to look at the molecule from the intact level and then perform the peptide mapping. This is like a proteomics experiment. Here I'm showing the UV trace. We use 0.1% formic acid additive in the chromatography. I will actually talk about this additive later. The reason we chose formic acid is because this is kind of a golden standard to help the sensitivity in the proteomics type of experiment. The product quality attributes we want to study include deamidation, isomerization, oxidation, truncation, and glycation. Today I will only talk about the 2. The mechanism of asparagine deamidation. It typically occurs through cyclic intermediate, could be isoimide or succinimide version. So it will have minus the mass shift. Upon hydrolysis, it can form either aspartic acid or isoaspartic acid. So the final product is about 0.1 Dalton shift. And it also can undergo deamidation through the side chain direct water-assisted hydrolysis directly. Deamidation typically increases with higher pH exposure and often occurs at flexible region. So as mentioned earlier, the general gold standard for running proteomics type of experiment is to use 0.1% formic acid. And we typically get really good resolution for deamidation analysis. However, as we started looking into more and more mAbs, we realized there are challenges with recovery or separation of hydrophilic peptides. And the practice we come up with to help mitigate the risk is to use 0.05% trifluoroacetic acid to help retain the hydrophilic peptide. However, there is one problem with this 0.05% TFA. It reduces the mass spec sensitivity. So really, we want to dedicate a certain instrument to this type of analysis. Because all our heightened characterization platform methods that I mentioned adopt TFA and maxis 2, it has all those merits we are looking into to help this analysis. We easily use maxis 2 as an add-on instrument for our hotspot analysis with the TFA chromatography. Here I'm showing you one example. This peptide contains 7 amino acids with the 3 potential deamidation sites. And this is the extracted ion chromatogram. The first eluting, so this peptide wouldn't be able to be retained with the 0.1% formic acid. With 0.05% TFA, now you can see we have unmodified peptide elutes early, and then followed by 4 additional peaks that belong to the deamidation species. All the species here are less than 2 ppm. So you can see this is an example showing that switching ion pairing rate simply from 0.1% formic acid to 0.05% TFA can allow retain of the hydrophilic peptide, and also all deamidation species can be separated here. Mentioned deamidation typically increases with a higher pH exposure. So now I'm showing data of the same peptide under stress condition. On the top is the control data, as I showed in the previous slides. And stress under pH 4.5 for 4 weeks. Now you can see there is a slight increase of deamidation. And at 5.8, there are a little bit more. Under 7.5, there's a significant increase of deamidation here. And this belongs to this NK motif. So this shows you that the deamidation at a higher pH occurs more often than stress under lower pH. And we always have orthogonal method to test dramatic modification. And as mentioned, we use ice and mass spec. Here I'm showing you a ice data. This is the ice trace, and this is the conditions on pH 7.5 at 2 weeks in the red trace and 4 weeks in the green trace. The amidation, because of the introduction of additional acidic side chain, so it will shift the species charge to the acidic region. So the significant increase of the acidic species indicates a high level of deamidation occurred here. And there is a good agreement between ice data and the mass spec data. Now let's talk about aspartic acid isomerization. In a similar way to deamidation, isomerization could also occur through the cyclic intermediate. The only difference is the final product, isoasp, has no mass shift compared to the initial aspartic acid version. And there is additional CH2 group be incorporated into the protein backbone, as shown here. So now you can think this additional CH2 group will introduce a kink into the protein structure, and therefore it may impact the protein function. So this conversion, unlike the amidation, typically increases with lower pH exposure. And the rate determining factors include primary sequence, environment, and the local structure. Challenges in this isomerization characterization. As mentioned earlier, there is no mass difference. Isoasp version versus asp. So to a mass spectrometrist, all we look for is the mass difference. So this is quite challenging. So often we rely on chromatographic separation for identification and quantification. However, sometimes we could get the false positive. Therefore, we need some not so common used fragmentation technology in order to unambiguously identify this modification. This ETD and ECD stand for electron transfer or capture dissociation. I'm not going into the details, but this is less commonly used in the mass spec world and may not be readily available to most of the labs. Just to demonstrate the challenges with separating isomerization, here I'm showing you an investigation on a few synthetic mixtures. So basically, for each peptide, I will order is either aspartic acid version or isoaspartic version, and I'm looking into the separation on 4 different columns. So you can see here on column 1 and 2, 2 isomers are well separated but not retained on column 3 due to the hydrophilicity, and co-elute on column 4. I will have a crying face here if the column is not performed as expected. And the second column I'm showing is a more complex example. This peptide contains 3 aspartic acid residues. So this mixture will have 4 different isomers. Isomerization at position 1, 3, 5, or the unmodified version. So on column 1, we have 4 species well separated. But on column 2, 2 of them co-elute. Column 3 and 4, the separation is quite good as well. So you can use the same analysis to look at the rest of the data, but the take-home message is even within the 6 peptide pairs I investigated, there is no single well separate them. So this is kind of alarm to us. What if this is a real-world case, and because of the selection of the column, we missed a very important modification? So the first strategy to mitigate that risk is to utilize diagnostic ions of electron transfer dissociation. Since I'm not going into the details, this is highly mass spec technical details. All I want to say is there is diagnostic ions, we call it Z-57, only present for the isoasp residues, but not asp. Both of these two -44 as a result of side chain CO2 loss. So here I'm showing you one. So this is a tryptic digest sample, tryptic digest of a mAb on one of the columns. So if I look at the full view, it's a single sharp peak, very nice. But if we zoom into this region, you have a very teeny peak elutes in front of the main peak. And also there seems like a shoulder, but it could be some column artifact. If we look at the ETD spectrum at these 3 different retention times, remember we're looking for this -57 as a diagnostic ion for isoasp. So at retention time 1, even if it's resolved at different retention time, we actually do not detect that satellite ion. In retention time 2 and 3, we have both of them will have this diagnostic ion, and at the retention time 3, the relative abundance increases. So this ETD data tell us that there are isomerization happening at position 2 and 3, but not at retention time 1. And it also seems like isoasp elutes slightly later than asp because the increasing amount of the -57 diagnostic ions. So okay, now we identify there are isomerization, but the quantification is prevented due to the chromatography challenges. So this leads us to trying to utilize the ion mobility separation. This is the Bruker timsTOF Pro instrument. And the concept, when I heard about the instrument, I was very excited about the ion mobility module because I want to see if this ion mobility could separate the isoasp versus asp pair. So the concept of ion mobility here is we can trap a few different ions in the gas phase because there is a push from gas, and also there is a resistance from the electric field. Depends on the shape, size, and charge of the molecules, they can be eluted out from the trapping device at a different time. So therefore, they have different mobility, and we can see if we can use that to separate over isomerization. So the following data are all from the direct infusion, And they were collected by Angela Giorgio at the Bellerica facility. So thanks very much to them. Here on the left, I'm showing you again the chromatography separation that I already showed a few slides ago. Just to remind us, the peptide pair containing aspartic acid and isoaspartic acid version. And on the right, I'm showing the team's data from direct infusion. This is the heat map. X-axis is M over Z, and the Y-axis is the mobility. Those different vertical, so the brighter the color is means more intense the ions are. So the vertical lines here just the different isotopic mass of peptides. And very clearly, if we follow the dimension of mobility, there are two groups here, which means that peptides traveling at two different mobility. And this view can be simplified if we look at the mobilogram. Converting from heat map to mobilogram, we just look at a certain M over Z, which is here, and we plot the relative intensity against the mobility. So now clearly you can see isoasp is separated from asp. We test with 3 other peptides, and you can see the length varies from shorter to longer length. And in all cases, isoasp is separated very well from the asp, as seen here. So now we want to challenge the ion mobility separation. We looked at a more complex mixture. So this peptide contains 2 aspartic acid residues. So we have 3 isomers in these peptide pairs. And here is the chromatography separation. So when we look at the mobilogram, there are only 2 peaks resolved. And the early eluting peak is a little bit almost like a double the intensities of the second eluting peak. So this suggests to us only 2 out of the 3 isoforms are resolved. An even more complex example, a peptide contains 3 aspartic acid residues. Isomerization at 3 sites, so the pair contains 4 different isomers. Again, here I'm showing the chromatography separation. On the heat map, there are only 2 distinct peaks. And the early eluting peak is almost triple the abundance of the later eluting peak. So this again suggests that only 2 distinct groups are resolved with this preliminary analysis. So it almost seems like isomerization at a different size cannot be resolved, but isomerization can be resolved from the aspartic acid. Just want to reemphasize here, I'm only showing the data using direct infusion, so means there is no chromatography separation at all in the front end. But imagine the separation power will be much more if we couple LC to the timsTOF Pro. So we are very excited to continue some of the studies here. And this is my last slide, second to the last slide. Just to recap what I talked today, I'm talking about a few case studies showing how we could advance the biotherapeutics characterization with ultra-high-resolution mass spectrometry. As mentioned, Bruker maxis plays a major role at our lab because it has really good versatility. We use the instrument for a wide range of assays and modalities. Today, I'm only giving the examples from monoclonal antibody, but we do for a few other modalities as well. The instrument is highly reliable. We can use it for rapid quantification of trisulfide, and it's just as good as compared to the gold standards, which requires more extensive sample preparation. And the resolution, this instrument has really high resolution compared to other Q-TOF in the market, and we are able to transform our platform's analysis from 3-part to 2-part. We have some internal data to show we can obtain monoisotopic mass for species up to 60 kilodalton. The mass accuracy is great. Less than ppm is routine, and it's really sensitive, and we also have semi-automatic workflow. We, Pfizer and Bruker, have collaborated for over 25 years applying state-of-the-art mass spec technologies for the heightened characterization of therapeutic proteins. That's why we continuously are interested in evaluating new technology. As shown, we evaluated the team separation in the timsTOF Pro for the isomerization case, and we see a very powerful third separation dimension here to assist our hotspot analysis. So in the end, I'd like to thank my colleagues at Pfizer, especially to Jason Ross, who is our Senior Director, and my manager, Olga Fries, and my other colleagues who have contributed data or discussion to this presentation. I also want to thank our hotspot analysis team, which is co-led by Jason and Laura Lin from another department. So we got to see a very, very interesting examples during this team. And finally, I want to thank Angela and Shoujo for collecting the data, and thank you all for the invitation for this presentation. And I'm happy to answer any questions.